Hello, everyone. So my name is Wouter Peelaerts. I'm a senior postdoctoral fellow in the lab of Professor Veerle Baekelandt at the KU Leuven in Belgium. And so my work and the lab's work has been focused on the role of alpha-synuclein strains in synucleinopathies. And so we've been doing this work for the last almost 10 years now. And so I'll be talking about the relevance and the importance of synuclein strains in these diseases. I also noticed that there are two other talks that also focus on the topic of alpha-synuclein and Parkinson's disease. So hopefully this talk will also supplement those other presentations as well. So as a quick overview here, so what I would just like to discuss is, first of all, the synucleinopathies, so just introduce these diseases, and then tell you why we think alpha-synuclein strains are important and relevant to these diseases. Then talk about synuclein strains and some of the experimental models and some of the experimental work that we have done. And then also finally, some words and some slides on how we believe the environment, more specifically the cellular environment, can also impact strain behavior and the development of these diseases. So the synucleinopathies is a large and also very heterogeneous group of brain disorders, so neurodegenerative diseases, and so it includes multiple system atrophy, MSA, dementia with Lewy bodies, or DLB and Parkinson's disease, or Ph.D. And what all of these diseases have in common is that when you look in the brain and the central nervous system, and even outside of the central nervous system, is that in the brain of these people, you'll find inclusions or aggregates of the protein alpha-synuclein. And so this is really a key histopathological feature that all of these diseases share. And so this reflects also this umbrella term synucleinopathies, reflecting the presence and probably also the importance of alpha-synuclein aggregation and pathology in these diseases. So alpha-synuclein comes in many forms and shapes, and originally was identified and initially characterized in Parkinson's disease, where it was found to be a key histopathological substrate in the disease. And so when people were examining, for instance, substantia nigra, which is a region that is heavily affected in Parkinson's disease, where there's lots of cell death, you find these protein deposits, inclusions within dopaminergic cells. And so these inclusions were then named after the person who initially described them, Frederick Lewy. And so then they were called Lewy bodies. So again, stressing the importance of alpha-synuclein protein here. So then when you look in more detail, into these inclusions, inside of these Lewy bodies. So here you can actually see a cross-section of how that looks like. You can see a cell here in panel A. In panel B and C, you can see higher magnifications of that inclusion. And you can see around like that big inclusion, lots of membranous and lipid accumulations, so reflecting organelles and mitochondria that are accumulating around what is a rich filamentous core of protein deposits. And you can actually, for instance, also purify or isolate the protein from these deposits, and you will see that you'll end up with these long insoluble filaments, fibrils, that are so typical and characteristic of the disease. So these are the synuclein fibrils that will have an important role in the disease process. So all of these diseases actually share synuclein aggregation as a main core feature, but there are also many differences, so not only in the clinical picture, but also on the cellular level. For instance, in Parkinson's disease and dementia with Lewy bodies, you'll find these synuclein inclusions within neurons. But then if you look at multiple system atrophy, you won't find the same type of, these typical inclusions in neurons, but instead you'll find them in glial cytoplasmic inclusions. So you'll find them in the oligodendrocytes, again, in the brain and even outside the brain, to the peripheral nervous system of people with multiple system atrophy. And this, for multiple system atrophy, again, is truly a core feature. So the inclusion of synuclein or the formation of synuclein aggregates within oligodendrocytes. And so because of this, Parkinson's disease and also dementia with Lewy bodies are primary neuropathies. So synuclein aggregation and pathology in neurons, resulting in dysfunctional neurons and eventually even neuronal cell death. So there you have a primary neuropathy. Whereas in multiple system atrophy, where you have the oligodendrocytes being affected, we more talk about a primary oligodendrogliopathy, where there's significant myelin loss, then resulting in a loss of trophic support, loss of metabolic support, and with that also a loss of neuronal health and neuronal cell death. These oligodendrocytes actually survive, and so you'll find lots of these inclusions, these gliocytoplasmic inclusions with synuclein aggregation, and so it's really the demyelination and the neuronal loss that again is a driving factor in the disease process of multiple system atrophy. So Parkinson's disease is by far the most common form of the synucleinopathies. 1 in 100 people above the age of 65 will have PD. For DLB, it's much less. It's not so well known what the exact prevalence is of dementia with Lewy bodies. There are some estimates, but it is considered to be less prevalent as Parkinson's disease. And MSA is a rare disease. It only occurs in 1 in 25,000 people, but it's also actually the most aggressive form of all synucleinopathies. So similarities, but also differences in the cellular level. And then when we look at clinical features, symptoms, and here I'm just going to focus on Parkinson's and multiple system atrophy, you'll also see some striking differences between these diseases. So here you have some of the core features of Parkinson's disease, and well, you can also map these here on a timeline. And what you'll see is that, so for Parkinson's disease here at the time of diagnosis, people will present with the more typical motor symptoms, neurology features that are so characteristic of Parkinson's disease. But even long before this, you will see that people have a multitude of symptoms that mostly associated with peripheral symptoms, such as constipation, REM sleep behavior disorder is actually an important prodromal symptom in Parkinson's disease, hyposmia, but even depression. And then once the disease advances, you'll have more symptoms adding on to the earlier features. And so that can also be mapped in terms of a spatial temporal pattern. And so normally I had an animation here, but just because of a technical glitch, this is not going to work. But what we're supposed to show here is that these synuclein protein inclusions can be found in different stages that correspond actually quite nicely with the pathology and the symptoms that people would exhibit. For instance, in terms of what we're going to call the Bragg hypothesis or the body first hypothesis, you could find these protein inclusions, for instance, first in the gut or in other peripheral areas, and then they would spread through either the vagal nerve or even sympathetic innervation through the to the brainstem, and then cause pathology and spread the pathology along anatomically connected regions. Next to that, there's now also the idea that there is a body first spatial temporal pattern where the pathology then initially originates in the nose or in the limbic system and then spreads in the opposite direction, resulting in an opposite staging scheme. So again, synuclein pathology correlating with the symptoms that people would exhibit or the regions associated with those symptoms. In case of multiple system atrophy, you immediately see that this disease has an incredible range of symptoms, and also here we can map all of these symptoms to a timeline. What you can see here is that the timeline, first of all, is much shorter. So instead of going up to 10, 20 years, people with multiple system atrophy usually live about 6 to 9 years with the disease. Even before diagnosis, you can see that there are many, actually very many premotor symptoms that characterize the disease. For MSA, typically people sometimes also develop urogenital dysfunction, so there's anorgasmia, erectile dysfunction, urinary incontinence, and so forth. Also, strikingly and interestingly, in this disease is the prominent infections that people will have and recurrent infections that they have, such as, for instance, UTIs. Autonomic dysfunction is also really prominent here. Again, REM sleep behavior disorder is also something that appears to be important in the disease process. And then again, later on, you'll have more and more symptoms that will be added on when the disease progresses. For multiple system atrophy, there isn't a typical spatiotemporal pattern that, for instance, would correlate synuclein pathology with clinical symptoms or pathology per se. But what we do know for multiple system atrophy is that there are two forms or two variants of the disease, where in one case, for one variant, the cerebellar functions will dominate, and in case of the Parkinsonian variant, there are more Parkinsonian symptoms that dominate. And in the end, people will usually converge, and symptoms will converge, and people will develop a full-blown MSA phenotype. So the synuclein pathology here with the glial cytoplasmic inclusions hasn't been mapped to the same extent as, for instance, the case of Parkinson's disease. So we don't really have good information on how the synuclein aggregation and the synuclein pathology correlates with the disease process. So that was the introduction to synucleinopathies. And so quite some time ago, our lab started investigating why there is only, or why there seems to be one molecular event in a large group of diseases, because we found it quite odd that you have this one protein, alpha-synuclein, and this protein being involved in so many different diseases, but still you would have diseases with such different phenotypes or such different clinical pictures. So we couldn't really understand how this one protein is involved in all of these diseases, and then people developing such striking different diseases. And the idea that we then had at the time is that synuclein itself might aggregate differently and form different types of structures. So the assembly into these filaments or the assembly into these fibrils would then be different, resulting in what we then call different types of strains. So reflecting the structural conformation of these protein aggregates. And we thought that if it is possible to, for instance, produce different types of aggregates, would it be possible that these different types of aggregates then would have different functions or different biological roles or different pathologies, and with that potentially cause, let's say, different diseases or different phenotypes. And so we started testing this, and we started working with the lab of Professor Melki, and in his lab was Luc Bousset, who was at the time generating different types of alpha-synuclein fibrils. And here in the left side of the left picture, you can see ribbon-type fibrils, which are aggregates or fibrils of alpha-synuclein that have this twisted conformation, and because they look like twisted ribbons, we call them ribbons. And then in the right panel, the right side of the picture, you can see just the more conventional, normal type of fibrils that are just rod-shaped and cylindrical. And so then we started experimenting with these two well-defined synuclein fibrils or synuclein strains, and then we wondered if we expose these and inject these in rat substantia nigra in an animal model that we had available at the time, would it be possible that these fibrils then would cause or trigger a phenotype that would reflect their structure? And so once we started doing that, we we noticed that indeed, upon injection of these different strains, the ribbons and the fibrils, that both strains were causing very distinctive phenotypes. For instance, here what is shown is just the histopathology of these fibrils when injected into the substantia nigra, with ribbons causing much more phosphorylated alpha-synuclein inclusions, which is the pathological marker that is used to detect these type of inclusions, so phosphoserine 129, the post-translational modification of alpha-synuclein that is found in deposited inclusion. And so you can see for ribbons that ribbons cause a much higher degree of inclusions and inclusion formation in vivo in the brain of these rats injected with fibrils. Then in contrast to this, when looking at neurotoxicity, interestingly, then we saw that these fibrils were actually causing more neurotoxicity compared to the ribbons. So even though these ribbons were associated with inclusion and inclusion formation, it was actually the fibrils that were causing more toxicity and cell death. And also, finally, interesting for this part, what we found is that upon injection of these ribbons in our model that was expressing human alpha-synuclein in our animal model, we found that these ribbons were also causing glial cytoplasmic inclusions, so the typical inclusions that you would observe in people with multiple system atrophy and oligodendrocytes, whereas for fibrils, we could not see this. So with those experiments, I think we showed that there is a relationship between the structure of a fibril or a strain and the phenotype and the pathology that will develop. So a strain A, for instance, then will cause phenotype A, and strain B will cause phenotype B. So that's actually the let's say the importance or the relevance of those strains in that particular animal model. So the next we asked, is it possible to do this with more, let's say, relevant brain material, because these were synthetic fibrils that were assembled in random buffer conditions. So one was assembled in, say, low salt conditions, whereas the other strain was assembled in physiological buffer conditions. So next we wondered, is it possible, for instance, to isolate these strains from patient brain and then again inject them into our animal models? And so what we did is we isolated synuclein aggregates from PD brain, DLB, and MSA brain. Then we incubated them just with synuclein monomers. We fractionated them by sonication, and then by templating the fractionated sonicated seeds of those isolated fibrils with new monomers, we were amplifying the material, and by doing this several times, by amplifying, so sonicating, amplifying, and so forth, you end up with pure homogeneous material that then again can be injected into animal models and can be studied. And so we did that for all of the three main synucleinopathies. So here we have 4 Parkinson's disease patients, here 6 MSA patients, and again 4 DLB patients. Here you can see the fibrils and the ribbons again. And so by amplifying, so purifying and amplifying those strains, we were now able to assess whether these fibrils isolated from human brain would behave the same way, so imprint their structural features into the environment to which they are exposed, and potentially cause different types of pathologies. So then again, by injecting all of these strains into rat substantia nigra, we found that the PD strains and the MSA strains both caused not only neurotoxicity, but also severe behavioral deficits. So here you see the quantifications for neuronal cell loss or dopaminergic cell loss in the substantia nigra. And here you can see some representative images of the nigra being gone, here in case for the MSA condition, PD, and then the control and DLB conditions next to that. But what also stood out here is that in case of the MSA strains or the MSA fibrils, that these MSA fibrils caused much more inclusions, again assessed by phosphorylated serine 129. We found that when injected in the substantia nigra, these inclusions would spread more throughout the brain, but also you would see much more of the inclusions when the animals were injected with MSA strains. So again, comparing them with the PD, the control, and the DLB material. We also saw the same the immune response, so looking at microglial activation, MHC1 staining, for instance, here, but also other markers for lymphocyte CD4 markers, peripheral immune cell infiltration, and so forth. We noticed that in case, again, for the MSA strains, there was much more inflammation after these strains were injected into our animal models. So in conclusion, for the strains, what we found is that synuclein strains can cause unique pathologies in multiple models. So we tested this in various cellular models as well as in our animal models. These strains, in fact, also amplicate and replicate in a strain-dependent manner, so you can isolate them back again from the brain, and then what you'll see is that they'll have replicated and amplified also in a strain-dependent manner. And also, finally, it's not only the work with the synthetic strains where we were able to show this, but also the work with the patient-derived strains from so relevant patient-derived material, where we found that, again, synuclein fibrils and these different alpha-synuclein strains can dictate different disease characteristics. And then just finally, to build on those findings, we've now been exploring what the role is of the cellular environment on strain behavior. And so I just have a couple of more slides on that as well. So as I mentioned earlier, there is an interesting cellular tropism when it comes to these different pathologies. So in case of multiple system atrophy, you will find glial cytoplasmic inclusions and accumulation of alpha-synuclein within oligodendrocytes, whereas in Parkinson's and DLB, these inclusions will form in neurons. Now, what is a bit odd is that when we take the strains derived from PD brain or strains derived from MSA brain and inject them in our rodent model, we find that after incubation and amplification and the pathology that they cause, that in case of the PD strain, you'll find a PD-like phenotype. But in case of the MSA strains, you'll find a sort of mixed phenotype where the pathology actually propagates within neurons instead of the expected oligodendrocytes. So there appears to be some sort of discrepancy here or dichotomy between the cellular environment and the strain that is amplifying. And the reason why this is actually came from a really interesting study by the group of Virginia Lee, where they incubated different types of primary cultures with synthetic forms, synthetic strains, but also patient-derived fibrils. And so after incubating, for instance, these fibrils here in a primary neuronal culture or in a oligodendroglial culture, both cultures expressing human alpha-synuclein, they found that after incubation and amplification of these strains, that in case of the neurons, they would end up with a slightly modified strain that would reflect the neuronal culture. But then after incubation here in the oligodendroglial culture, they would find that the strain would have much more modified, much more infectious, and much more toxic suddenly. So it seems that an oligodendroglial culture or an oligodendroglial environment is able to have an effect or modify the amplification and the propagation of synuclein fibrils or synuclein assemblies. And so to investigate how this really works, we also started working again with the ribbons and the fibrils shown earlier, but instead of using a, let's say, more Parkinson animal model, we now switched to a multiple system atrophy model. So the mice that we used for this experiment are transgenic mice that express alpha-synuclein under control of the PLP promoter. And so by that, they express alpha-synuclein in oligodendrocytes. So now we have an environment that is prone to develop multiple system atrophy. And so we wondered, is it possible that these strains can not only, let's say, impose or imprint their structural features and their function within the environment they're exposed to, but also is it possible that they can develop and contribute to the MSA phenotype? And so after a period of 11 or 12 months, we then analyzed the substantia nigra striatum and the different brain areas associated here with basal ganglia. And so what you can see here, for instance, immediately, if you look at panel J or image J here, this picture, is that these fibrils cause tremendous brain atrophy compared to ribbons, where the effect is even not there. And in any assay that we used, any assay looking at cell function or cell death, we found that these fibrils were, again, the most toxic species, causing significant dopaminergic cell death, loss of DARP32 medium spiny neurons in the striatum, loss of total number of neuronal cells, again here in the striatum, also very striking loss of myelin, so demyelination here as well, and again loss of striatal volume and an increase in ventricular volume. So these fibrils again are causing very significant neurotoxicity, whereas these ribbons do this to a much lesser extent. So then next, what we wanted to assess here is how these strains and the environment then are, interacting. And for this, we started working with luminescent conjugated oligothiophenes, or LCOs. And more specifically, we started working with HFTAA, which is one of the many LCOs that are available. And so these are developed by the lab of Peter Nilsson, and so we've also been working with Therese Klingstedt, who's been very helpful in setting this up here in our lab in Leuven. And so these LCOs are flexible fluorescent molecules that can bind with the amyloid, and in case of the hepta-FTAA LCOs, that can bind with anything that is as big as 7 monomers. Anything larger than that can be bound by these probes. And so the interesting thing about these is not that they're just very specific and sensitive. For instance, they're much more sensitive than, for instance, thioflavin, but they also have their own spectral signature. So they can bind with different types of assemblies, and because of the backbone being flexible, they can also bind with different types of assemblies and have a different spectral signature that will then translate to the conformation of these different types of fibrils. So for instance, if you have it bound to a ribbon or a fibril, you will see a different spectral signature. And so as a way to set this up in our lab, then I started just staining first some MSA brain and some control non-diseased brain, just to double check how the probe would bind with glial cytoplasmic inclusions, and to see if we could actually use this for our animal models. So on the left, you see staining for phosphorylated alpha-synuclein. Staining for phosphorylated alpha-synuclein. So in the control sample, it's negative, and for here, this person were stained to caudate putamen. You can see lots of inclusions in glial cytoplasmic inclusions, indicative of synuclein accumulation and also multiple system atrophy. So then when combining this marker with the LCO probe, you can see again that this is negative in the control condition, but then for the MSA condition, you can see that there's a nice overlap and correlation between the two markers. So showing that indeed we can use this to look at the glial cytoplasmic inclusions in our not only patient samples, but also in our model. So this is really to illustrate I think the real beauty of this technique, because you can combine this with other markers. So here you can see OLIG2, which is a marker for oligodendrocytes, and with that you can go looking specifically for inclusions in oligodendroglial cells. And by doing so, then I was able to determine the emission spectra here of the probe bound with aggregates inside oligodendrocytes. And so I did this for all of the different conditions that we had here. So just the transgenic models, so these were animals injected with BSA, then the animals injected with ribbons, the animals injected with fibrils, and then also the MSA patients. And so what you see is that for all of these different conditions, you will have different emission spectra and different types of spectral signatures. And then this can be quantified, and with these quantifications, you can see that in case of the BSA conditions, you have this very variable and heterogeneous folding landscape. So you can see the data points really stretched out along the y-axis, again indicating that there are many different types of conformations in this model, and also that there isn't one particular, let's say, dominant conformation in the transgenic model itself. But then when you, for instance, inject the fibrils, you can see a shift in the conformational state of these oligodendroglial inclusions. And even more so when you look at the ribbons, you can see that these ribbons are, again, imprinting their structural state within the cellular milieu or cellular environment where the propagation is happening, and also that it is reflecting more that of the multiple system atrophy cases. So I'm just going to skip these couple of slides and just go directly to the conclusions because I see I'm running out of time here. And so with that, I think I was able to show that there is an important correlation or an important relationship between strains and the environment. So oligodendrocytes can clearly impact strain propagation, where the environment is really a facilitator for, in case of multiple system atrophy, and a phenotype that develops here with providing the soluble synuclein or the synuclein monomer for these strains to amplify. So the phenotype here in this case is actually determined by the strain and the cellular milieu. And so by seeding in our MSA model with different types of fibrils, we are able to more closely develop an MSA model and again the MSA phenotype. So that just leaves me with the last slide here, which are the acknowledgements, of course, for the lab in Leuven, the lab of Professor Veerle Baekelandt and everybody who contributed to this work. Also Ronald Melky, who's been really instrumental in all this work, Peter Nielsen also as well here, and Nadia Stefanova for her help with the transgenic MSA model. And of course, everybody here attending this presentation. So thank you for your attention.